Forensic Science Lecture 12-3, Forensic Examination of Fibers. Forensic examination of fibers become important evidence involving crimes of personal contact. So as of Lockhardt's exchange principle, the fibers will exchange between the victim and the suspect when they come in contact with one another. The significance of the fiber evidence, however, is dependent, much like that of the hair. It's the criminalist's ability to narrow down the origin to a limited number of sources or even a single source. However, due to the mass production of fibers, there's limited value in fiber evidence, and it's more rare that fibers recovered can provide individual identification with a high degree of certainty. Most of the time, it comes into class evidence. We have two types of fibers: natural fibers, which are derived only from animal or plant sources, so wool, cashmere, fur, cottons, and manufactured, which are man-made: rayon, nylons, polyesters, acrylic, and so on. When we're looking at natural fibers, these animal fibers use the same procedures for examination as hair fibers. They rely on microscopic examination for color and the morphology of each fiber. The most prevalent plant fiber that's used in fabrics is cotton. So white cotton in clothes and other fabrics is almost meaningless because they all have the same appearance. However, when they're dyed, this enhances the value, and it's really the dye of that fiber that is providing the evidence of comparison. Cotton, though, is distinguishable by a feature that has a ribbon-like shape with twists throughout the thread, and so this is easily identified as cotton. Manufactured cottons have two types: regenerated fibers, and these are manufactured from natural raw material, so rayon, acetate, triacetate, and then synthetic fibers, which are the most commonly manufactured, are produced solely from synthetic chemicals: nylons, polyesters, acrylics. In synthetic fibers, the production base of these fibers uses nature's principles, the same principles that nature uses to make hair in vegetable fibers, but it's done in a lab. They take the polymer or the macromolecule, which is the synthetic fiber, which is composed of a large number of atoms arranged in those repeating units, which are monomers. So by taking the monomers and linking them together, they can create polymers of these synthetic fibers, the first of which to have been done was nylon. And typically, as most everything, it was accidental, but as they pulled the nylon polymer through a small hole, it became more and more flexible yet sturdy. And so that was the start of synthetic fibers. When we look at identifying and comparison of these manufactured fibers, again, the value is going to lie in the ability to trace the origin of the fiber. The first step is to use a comparison microscope for the color and the diameter. Other features they look for: length-wide striations, pitting on the surface. This is made with a delustering particle, usually titanium dioxide, and it's what manufacturers add to reduce the shine of the fiber. They also can look at a cross-section shape of these fibers that can help to characterize it. The second step in comparison is dealing with the composition of the fiber, the chemical makeup. Although two fibers may appear to have the same color, a compositional difference may exist in the dye that were applied during the manufacturer process. And so they need to take that and come up with the chemical composition of that dye to help identify. Other analytical techniques that use to examine fibers is a visual light microspectrophotometer. And this is a convenient way to compare colors of fibers through spectral patterns. It's not limited by sample size, and it's actually non-invasive to the sample, so it's used often. But a more detailed analysis of the dye composition can be obtained through uh, chromatography separation. And so the dye is extracted from the fiber, and then the thin layer chromatography is carried out and compared side by side. Another analytical technique that they use is infrared spectrophotometry and polarized microscopes. And these are rapid and reliable methods for identifying a generic class of fibers. So depending on the class of fiber, each polarized plane of light have a characteristic index of refraction. And there's a reference database for that. Unfortunately, though, there's no analytical technique that prevents the association of a fiber standard definitively to a single garment, and there's no database that exists from determining the probability of the fiber origin. Um, of course, if analysts are able to link two or more fibers to the same object, that's going to increase the significance. But even so, there's an enormous variety of fibers that exist in our society, and it's unlikely that two different people are wearing identical colored fabrics, with, of course, the exception of blue denim and white cotton. But by comparing color, size, shape, microscope appearance, chemical composition, and dye content, it can have significance of evidence for the investigator. When we're talking about collecting and preserving fibers, fiber evidence can be associated with, again, almost any type of crime, just like hair. However, it can't be seen with the naked eye and is easily overlooked. Investigators are tasked with identifying and preserving potential carriers of fiber evidence. Any relative articles of clothing should be packaged carefully in separate bags. Again, anytime we're packaging any sort of evidence, it needs to avoid contamination at all costs. So wearing the proper gear, using the proper tools. If at any point it's necessary for fibers from an object to be removed by the investigator, they need to use clean forceps, place it in a bindle and label it, and then the bindle typically is placed inside of another container with the chain of custody attached to it. This ends chapter or lecture 12-3. There's a quiz next class.